One of the most promising new treatments for brain tumors is a vaccine that can stimulate the immune system to attack and kill cancer cells. In this video, we will explain how this vaccine works and what are its benefits. The vaccine is called Cervax-M, and it was developed by researchers at Roswell Park Comprehensive Cancer Center in New York. Cervax-M targets a protein called survivin, which is found in high levels in many types of cancer cells, including glioblastoma, the most aggressive and common form of brain tumor. Survivin helps cancer cells survive and resist chemotherapy and radiation. By blocking survivin, Cervax-M aims to make cancer cells more vulnerable to treatment and more visible to the immune system. Cervax-M is given as a shot under the skin, usually in combination with standard therapy. The vaccine contains a synthetic version of a part of the survivin protein called SVN53-67 attached to a carrier protein called KLH. This combination triggers an immune response that produces antibodies and T cells that recognize and attack survivin-expressing cancer cells. Cervax-M has shown promising results in clinical trials involving patients with recurrent or newly diagnosed glioblastoma. In a Phase 2 trial of 63 patients with recurrent glioblastoma, Cervax-M was found to extend survival time to 26 months on average, compared to 15 months for historical controls. The vaccine was also well tolerated, with mostly mild side effects such as injection site reactions, fatigue, and muscle pain. Another Phase 2 trial of 50 patients with newly diagnosed glioblastoma is ongoing, and preliminary results suggest that Cervax-M may improve survival and delay tumor progression when added to standard therapy. The researchers are planning to conduct a larger Phase 3 trial to confirm these findings. Cervax-M is not the only vaccine being tested for brain tumors. Another vaccine, called DCVax, uses dendritic cells, which are specialized immune cells that present antigens to other immune cells. DCVax is made by taking a sample of the patient's tumor in blood and then exposing the dendritic cells from the blood to the tumor antigens in the laboratory. The activated dendritic cells are then injected back into the patient, where they stimulate an immune response against the tumor. DCVax has also shown encouraging results in clinical trials, but it has some limitations. For example, it requires surgery to obtain the tumor sample, it is more expensive and complex to produce, and it may not target all the relevant antigens on the tumor. Cervax-M has some advantages over DCVax, such as being easier and cheaper to manufacture, being more consistent and standardized, and targeting a universal antigen that is present in most glioblastoma cells. Both vaccines are examples of how immunotherapy can offer new hope for patients with brain tumors who currently have few effective options. By harnessing the power of the immune system, these vaccines may be able to improve survival and quality of life for these patients.